Hello everyone, welcome to biology classes. I am Ishita Sharma, and in this video we are going to discuss about red blood cells. So in previous video we discussed about composition of blood, in which we discussed the plasma, what is plasma and what are its functions. In today's video, we are going to discuss about formed elements or blood cells which constitute 40 to 45% of the blood volume. So let's just discuss the types of blood. So basically there are three types of blood. The first one is RBCs or red blood cells. These are also known as erythrocytes. So right, we here in this picture we can see that erythrocytes. Next is WBCs or leukocyte, white blood cells or leukocyte. Here we can see the WBCs. And the third one is platelets or thrombocytes. We can see platelets over here. So let's just basically discuss what are blood corpuscles. So these are the cells which freely floats in the plasma. It constitutes about 40 to 45% of the blood. The percentage of blood cells is called as hematocrit value. So how much blood is present in a given amount or a fixed amount of blood, it is known as hematocrit value, or also known as packed cell volume. Packed cell volume. So in male or a human male, it is 40 to 45% the value of hematocrit, and the hematocrit value in female is around 36%. So this packed cell volume, it is measured with the help of an instrument which is Winthrop's tube. Winthrop's tube. So here in this picture you can see the Winthrop's tube. So this figure is the Winthrop's tube, by which we can calculate or determine the hematocrit value. So let's just discuss about the first formed element, that is RBCs or erythrocytes. So RBCs, as the name suggests, red blood cells, because it is red in color, hence it is named as red blood cell. Let's talk about its shape. So if we talk about its shape, it is different in different organisms. In vertebrates, this is a vertebrate. In vertebrates, it is oval, biconvex, and nucleated. In Only in mammals, nucleus is absent, but in earlier forms or the young forms of these RBCs, nucleus is present, but in matured stage, uh, it is not present. The nucleus is not present. In mammals, RBCs are circular, as we can see over here. These are circular, biconcave. And why the shape is biconcave? What is the advantage of this shape? So biconcave shape increases the surface area. It increases the surface area so that oxygen can easily diffuse in the RBCs, and more oxygen is transported from RBCs. So nucleus is also absent in the matured stage of mammalian RBCs, because the hemoglobin content. Hemoglobin is the pigment that is responsible for oxygen transport. So this pigment is helpful in oxygen transport, and if nucleus will be there in cell in a RBC, it is more difficult to transport the oxygen and less hemoglobin content. So there are some exceptions in mammals, in which the cells contain nucleus and also oval shape. These are not biconcave shape. So there is a special condition that is known as spherocytosis. Spherocytosis is a condition in which RBCs lose its biconcavity. So it is that leads to the less dissociation of the oxygen. So let's just discuss about the size of RBC. In humans, the diameter of RBC is around 7.5 micrometer. So around, as we can see in this picture here, its thickness is around 7.5 micrometer, and at the edges the thickness is around 2 micrometers, and in the center, it is not mentioned in this picture, the its thickness from the center is around 1 micrometer. So among mammals, the elephants are the ones which have the largest size RBCs, and in vertebrates, if you talk about vertebrates, amphibians are the one with largest size RBCs, and the smallest one is of mouse deer. So volume of human RBCs is around 90 to 95 microcube, and the surface area is around 30.5 microsquare. So what is the number of RBCs in a human body? So the total number of RBCs per liter or microliter of the blood is called its RBC count, and the RBC count of male is around 5.0 to 5.5 million cubic meter of millimeter of blood, and in females this value is around 4.5 to 5.0 million cubic millimeter of the blood. So there is an instrument here, we can see this. This is the instrument. Let me change the color, please. So this is the instrument over here. This instrument, it is used to detect or determine the RBC count, and the name of this instrument is hemocytometer. So the instrument used in RBC count determination, it is hemocytometer. So there are some factors through which the number of RBCs can be increased or may be decreased. So these are two factors. First factor in which the RBC number increases. So there are two factors to this. First is the physiological state. When we perform any exercise, the demand for oxygen increased, and hence the number of RBCs also increased. And if we are in higher concentration or a higher altitude, like in the air, the oxygen concentration decreases, or there is low oxygen concentration, and in that reason the RBCs are created more in number. And decrease in RBC, it is due to hemorrhage or hemolysis, that cause anemia. So decrease in the level of RBCs cause anemia, and increase in the number of RBCs cause polycythemia. Let's just discuss about these two diseases. So first one is anemia. So the decrease in number of RBCs or a decrease in RBC count is referred to as anemia. So what are the reasons that is responsible for causing anemia? So these are some of the reasons that includes blood loss, ulcer, cancer, piles, iron deficiency, vitamin B12 deficiency, hemolysis, or any parasitic infection. So there are two types of anemia. First is megaloblastic anemia, that occurs due to the deficiency of vitamin B12 or folic acid, and another one is aplastic anemia, that occurs due to the depression of erythropoiesis in the red bone marrow. Uh, and anemia leads to hypoxia or oxygen deficiency in the body tissues. That's why the anemic patients are always restless, वो बहुत हमेशा थके हुए रहते हैं. And as we can see in this picture, so this is the normal red blood cell count or RBC count, and in anemic patients the number of this RBC is less. This is anemic red blood cell count or anemic RBC. Now let's just move to the another one, that is polycythemia. So polycythemia is when the RBC count increases more than normal level in the body, then it leads to the disease called polycythemia. So this polycythemia can be primary, and when it is primary, it occurs due to the cancer proliferation stem cells, and cancerous proliferation of stem cells. When stem cells there is cancer causing cells. These are some cancer cells like this. And secondary polycythemia occurs due to some other diseases like emphysema or some pathological conditions like higher altitudes, because number of RBCs in higher altitudes is increased. Let's just see this image. The polycythemic patient has the more number of RBCs than normal level. As we know that We want adequate number of RBCs in the body. So if there is low RBC count or a high RBC count, both are really injurious to our health. Uh, Now here comes the color. So the color, as the name suggests, RBCs have red color, but it is not always there, because when the RBCs are present in singly, when there is single RBC, the color is yellow, and when it is present in bulk amount, then it appears to be red. So why the hell this RBC is red in color? The reason to that is the pigment that is known as hemoglobin. So hemoglobin is a iron containing pigment that provides the red color to the RBC, which is present in the cytoplasm of the RBC, red blood cell. So this is the hemoglobin. This is the structure of hemoglobin, as we can see over here. This is the structure of hemoglobin. Here is iron. This is heme group. There is beta chain and there is alpha chain. We will discuss in next video about the whole structure of hemoglobin. So let's just move that. I forgot to tell you that Rolex formation. So RBCs undergo Rolex formation. So what exactly is Rolex? So when these RBCs move slowly, they have the ability to stick over one another, and this sticky nature forms a complex that is called Rolex. In this slide we can see that we can see that RBCs are sticking one over other. So this is the Rolex. This is the Rolex. This exactly is the Rolex. So let's just discuss about the functions of hemoglobin. First is oxygen transport. So hemoglobin is responsible for all the oxygen, 97 to 99% of oxygen that is transmitted from lungs to various parts of the body. And the hemoglobin form along with the oxygen form an oxyhemoglobin complex. As we can see in this picture, this is oxygen plus hemoglobin and it forms oxyhemoglobin, which appears bright red color, right? And one mole, the one molecule of Hb or hemoglobin can carry 4 molecules of oxygen at high partial pressure of oxygen, and in 19.80 ml of oxygen is transported by 100 ml of blood. So one RBC can transport in its lifespan, it can transport around 1120 million oxygen molecules. Because of this hemoglobin, it has high affinity for oxygen, right? Like this. The hemoglobin attaches to the oxygen like this, as we can see in this image. Apart from oxygen transport, hemoglobin also plays an important role in carbon dioxide transport. In this picture, we can see that. And when it transports carbon dioxide, it forms carbaminohemoglobin. Carbaminohemoglobin, which appears to be dark cherry color. And it is transported from body tissues to the lungs for oxygenation. Next is pH consistency. It also acts as a buffer that helps in maintaining the constant pH. So that was all about the RBCs. In the next video, we are going to discuss about structure of hemoglobin and erythropoiesis. Thank you so much. If you really like this video, don't forget to subscribe the channel and click the bell icon. Thank you so much.